Hello, and welcome to this video tutorial of plate number 94, the eye. In this video, I will talk about the basic overall eye anatomy, and then a little more specifically about the retina, all of the different specific cells and layers of that very sensitive retina. Okay, so I am on plate number 94. If you want to have that opened up, my image will look very similar. I did pick one in black and white. I think it's a little easier to kind of see. There are a bazillion images out there. If you take a look at the eye with their varying level of complexity. I'm going to do an overview and kind of talk about the parts that you would want to color if you are going to color plate number 94, which is optional, but if you feel it will help you out, then please do so. Overall, the eye is, I think it's about 2 centimeters in length. We'll talk about in this section many different functions, or many different problems that can happen with the eye. We'll talk about how the eye actually works. We'll talk about if your eye is a little too oblong. So Mr. Eckert's eye was a little bit too long from the front to the back. That caused him to not be able to see far away. In some people, their eyes are too squished together, so to speak, I guess you would say. So their eyes are a little more flattened, which would mean that this would be compressed. So the distance from the back of the eye to the front of the eye is a little less than expected. And so we'll talk about how you can do corrective vision and so forth. So let's go through the anatomy. I'm going to kind of start on the outside and work in. So layer A is called the sclera. That is the white of your eye. So when you can see kind of the white of your eye, that is the sclera. I know it's probably difficult to color that in white, and I'm not sure if the coloring notes have you do that in white. But anyhow, so the sclera is the white of your eye. The next layer we would come to is called the choroid layer, not to be confused with the coronoid or the coracoid process. This is just the choroid. This is a layer that has lots and lots of blood vessels that will be feeding the retina, which is right below. So you can see how they've divided this up. So the sclera is the outside, the next layer would be the choroid, and then the retina. The retina lines the inside of your eye from about right in here. You can kind of see in your eye. And you have to remember, this is three-dimensional, so this is a little deceiving. But the retina is very much the back of your eye that the doctor may look at if he or she, if you're at the eye doctor and they look through your pupil and they are looking at the back of your eye, they're actually looking at these blood vessels that are back here. They're making sure that those are all nice. Folks that have diabetes, one of the early symptoms of diabetes is actually these blood vessels start to become damaged because of the blood sugar. When you have high blood sugar a lot, actually damages very tiny blood vessels, and that's one of the first things. Early detection is actually eye. Good eye health can catch diabetes more quickly than some of the other symptoms, especially for that type 2, adult diabetes, whenever you're getting it when you are an adult. Next, let's talk about the ciliary body and the ciliary process. Let me actually first talk about the lens. The lens is right here. This is going to be letter J. It tells you not to color that. If you want to color that, that's fine with me. If you're coloring, there really aren't any rules when you're coloring. Now, that lens has to be able to change shape, and that's one of the things when your eye is focusing. This lens must get pulled, so it's a little thinner, and then sometimes it needs to be relaxed, so it's a little thicker, and that's going to what focuses, or partly, it's half of the focusing process on your retina. Attached to that, that are attaching your lens to the body of your eye, be part of the sclera and all those different parts. It's called the suspensory ligaments. Now, this is not a ligament like when we think of ligaments, we often thought of bone to bone. So this is a little different kind of a ligament. There are no bones here. And it's called suspensory because it really is suspending up and down. It is suspending your lens. You have to remember, all of this is actually floating in liquid. The inside of your eyeball is a clear liquid. And so we have the lens, the suspensory ligaments. Then we have the ciliary portion. There is the little part that's sticking out here, and your book labels this, and you can probably tell this better in your book. This is the suspensory process that kind of sticks out. That's what the ligaments would attach directly to. Let me get a better pointer here. So you have the ligaments. Then here is the suspensory process. So I'll kind of color it in blue. So there's my process, and there'd be a process down here. It's just the end of that where those ligaments would attach. Then we have what's called the suspensory body. So the suspensory body, and they do that CNC1, which is usually the same color. If you want to do it in different colors, that's fine with me, again, if you're coloring. And so that portion is just sort of this, and it's actually a muscle as well, because it's going to contract when the lens needs to be kind of stretched out a little bit. So there's our suspensory process. All right, so we've done the sclera, that's the whites of your eye. Oh, I skipped over the cornea. The cornea, so your sclera, eventually when it gets to the very front of your body, actually becomes a clear set of cells. The cells, and this is interesting, it's a very unique patterning. The cells are most crystallized. We generally don't see clear cells, but running, kind of do this in pink, and there's, I don't know the distinct area, but so here all the way over to here, these cells here that make up your cornea is clear for letting for light passing through. The light passes through, and then you have, we'll get to that in a minute. We'll talk about your pupil. Now, on the very, 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 very outside, of your, I make that too. We'll do this in sort of a yellow color. The very outside of your cornea, you have a layer of cells called the conjunctiva. The conjunctiva. They're just a very, very thin layer of epithelial cells. Those do the covering. And this is where, if you've ever heard of pink eye is technically called conjunctivitis. So the reason it's called conjunctivitis is because you have inflammation in that outside layer. You have swelling, itis, in that outside layer. All right, let me clear some things here. Next, we have the iris. So the iris is going to be the part that's colored of your eye. So let's make this person have blue eyes, since Mr. Ackert has blue eyes. So this little area here is a muscle. It is actually rings of muscle. You have to remember this is going around, so this is only one-dimensional. And these are the ones that can contract. They can dilate. Dilate would mean they open up, and if they constrict, they contract and they get smaller, and that controls your pupil. So your pupil would run, really it's any place where there's not iris, so the pupil is just a hole. It's actually nothing, which light can then pass through. So again, if your iris contracts, actually, if it contracts, it actually gets smaller. The pupil gets smaller because the muscle itself actually kind of enlarges. It's a radial muscle, kind of works interestingly. It doesn't pull and push. It's a ring of muscles. And then if it relaxes, it actually opens up and lets more light in. So your pupil, and it actually says you can't color. It tells you not to color your pupil, mostly because it's an empty spot. So it's a little arrow with a little black dot on it, which means, or you could color that in black if you want to color the little arrow in black, so you would know. All right. So I've got the iris. We've got the retina. Remember, lines the inside of your eye. Now, the very back of your eye, back here, where all the blood vessels actually kind of merge, and and also this is where the nerve. So you're going to have a nerve back called the optic nerve, kind of all converge and exit the eye. But this is called the optic disc. The optic disc is called that because when a doctor looks in the back, it does kind of look like a rounded area, disc shape. Now your eye is, as I mentioned, in fluid. So anything from the lens back, this is called vitreous humor. Humor is a fluid. So the vitreous are back here. And then in the front, behind your cornea and in front of the lens, and these are almost two different chambers. Fluid can flow back and forth, but you almost think of them two different chambers. This is called the aqueous humor, and I can remember the aqueous because it's in the anterior of the eye. If you remember so anterior and posterior. So the aqueous is in the anterior of the eye. Okay. We've talked about the lens. Again, you may color that. We've talked about suspensory. The optic nerve. So leaving your eye, heading to the brain back here. we would have embedded in the optic nerve. So this is the optic nerve, and I think they ask you to do that in yellow. I think nerves are generally in yellow. You would have blood vessels that would be feeding and taking blood away from the eye. So the red that I've done here, that would be the optic artery, or yeah, the optic artery, or what they call the retinal. The book may actually call it retinal. Yeah, it does call it retinal. And so really, because we're just feeding the retina of the eye, it needs lots and lots of blood. Not compared to your brain, but a good portion of blood. So the red would be taking blood to the retina. The blue would be taking blood away back to the lungs to pick up oxygen and along the way pick up new nutrients. So we have the retinal artery and its branches, and in your text it kind of breaks it down into whenever the artery kind of gets smaller and smaller branches and branches, and then the retinal vein, which they'll ask you to do in blue. All right, so that's the overall eye. Hopefully that was helpful. Now let's talk about the we're going to get in detail here. We're going to zoom in on the retina and its different layers. Now it's important to understand where light would be coming in here. So in this drawing, because it does look kind of crazy, it's on a cellular level, but it's important to understand that light would be coming in. So let's do some yellow for some, that's sort of yellow. That's sort of yellow. So out here's the sun. Whoop, here's our sun. And so light is passing into the eye this way. So the light will actually pass through some of these different cells to get down to this layer here. And we'll talk about that layer in just a moment. And I am going to work from bottom up here. So this is a kind of a strange depiction of the sun, but it is important to understand that the sunlight does pass through these layers and then hits these cells that are down here. These are the cells that are going to be gathering the light data. So we talk about, so down here would be the, so, and this is extremely thin. The choroid would be thicker than this, but I also just wanted to point out that down here we would have the sclera of your eye. Down here would be the sclera. Oop, that wasn't very good to see. So sclera, the choroid layer is feeding the retina. It would be very vascular, so it's going to feed that layer. All right. Next, we have what's called the pig. So I'm going to kind of work up. So we've got, so the sclera was down here. The choroid with its embedded blood vessels, and I think it asked you to color this in and there'd be some little blood vessels that would be periodic because it is feeding the retina. Next, the pigmented epithelial layer. And in your text, it kind of does layers, and then it also does the cells. So these are the pigmented epithelial cells. Now, because they're called pigmented, doesn't mean this is where you get the color from your eye at all. It is a bit deceiving. The idea, they're called pigmented because they're actually producing some pigments that will move up into these cells for picking up photoreceptors or picking up light and different things. But this very thin layer, it's only a single layer, and it is just actually that. These pigmented epithelial cells and layer. So P, nice they did P for pigmented. Next, we have this photoreceptor layer. Now, the photoreceptor layer, you actually won't necessarily color this layer. And so it just says, realize that this is, and so I don't know if I have in my purpose game. So this layer here, this is the pigmented I'm on pigmented still, photoreceptor layer. Called photoreceptors because there are two kinds of cells that are going to be receiving sunlight and take that light data. It doesn't have to be sunlight, it could be any kind of light in a room or whatnot, but taking that light data and processing it. There are two types. We have rod cells and cone cells. Rod cells, it's important to note, and we'll talk about this later, these are only sensing the presence of light. These can't differentiate between what color it is. They are only going to be seeing things that there is light or there isn't light. Rod cells are your night vision. So actually in the dark, if it's a low light, you actually only see in black and white. People don't necessarily think of this. Now, whether that's in different, it will depend on how bright the light is before you start seeing color. But if you are in a dim room or a dim closet or same place dim, you only see in black and white, which is, you may not have thought of that before. There are more rods than there are, so rods they do in purple here. There are more rods than there are cone cells. I think the ratio may be like 10 to 1 or 100 to 1. It is different in different parts of the retina. Um, when you color those, maybe it actually has you do them in gray. The asterisk means in gray. That kind of reminds you that those are only seeing in grayscale. Then this picture has 3. Your drawing also has 3. It's O2. And these are your cone cells. These are what are processing light. But not every cone senses light the same way. Cone cells will have a special pigment that will see a range of colors. Generally speaking, there are cones that we'll see in the blue range, so the blue spectrum. And again, that's a range of things. So those will be seeing purples, and they'll be seeing, it's a range in the kind of the blue family. Then there are some that we'll see in the green range, and this would include the yellow. As it turns out, blue and green, partway through there, that's your yellow spectrum, if you remember Roy G. Biv. They would be seeing that range, and then there are cones that are red for seeing in the red ranges. Remember, because light is a color spectrum, it's a different set of wavelengths, it's a set of wavelengths and whatnot that we can see. Now, I may recommend that in your picture, or if you're thinking about this, that you do one in blue, you do one in green, and you do one in red. Now, I realize that red you would have used for the arteries, but that's okay. Just kind of a way of kind of remembering that some would see in the red range, some would see in the blue, and some would see in the green. And that's where color blindness comes in. If somebody is lacking the blue cones, I had a student a few years ago that didn't have any of his blue cones. He could not see the color blue. That was a pretty rare color blindness. More commonly are the green or the red color blindness. Those are the ones that are more common. So those are our rods and cones. Those are our photoreceptor layer. Moving up, so they're going to take that data and they're going to pass it on to this next set of cells. And that will tell you there are some cells that also are involved with this that we'll see later. Sometimes there are some cells called horizontal cells that will actually be passing information back and forth. Sometimes these are collecting information on if you need to if you're getting enough light to open your pupils up. There's a specific cell called a horizontal cell, which kind of works side to side to see if you do have enough. If you have enough light, and if you don't have enough light, they'll open up your pupil, and if there's too much, they'll close it down. But this next layer is called the bipolar cells or layer. So here's the bipolar layer would run all of these cells. These bipolar, if you notice, they're actually gathering. So this particular one is gathering 1, 2, 3, 4 different rods and combining that information. So they're gathering and kind of putting information together. Here is another one of our bipolar cells, and it's gathering from 2. And if notice that almost all the rods are combining information by these bipolar. However, when you have these bipolar cells, generally speaking, they're attached to much fewer color of the cone cells. And so this is so we can actually see to a very high acuity. We have very sensitive eyes when it comes to light in good conditions, and can see things in very great detail, whereas in black and white, at night vision, you don't really need to be able to see images that clearly. You just need to see the presence of those. So by combining many of them, you have better night vision that way. So cats that can see really well in the black in dark conditions, their eyes would even have more of these rod cells. Color isn't as important for them. All right, so we've got our sensory layer here, our photoreceptive layer. Then we have our bipolar cells and bipolar layer. Then we have these cells called ganglion cells and ganglion layer. These are what are starting to form. These are starting to form the optic nerve. So all of these things. So the way a nerve is put together is it has a body and it kind of receives information on things called dendrites. And then it has an axon. Axon is sort of how it transmits. It's a cable that helps it transmit the information. So these start getting bundled together to make that optic nerve. And in your picture, you can kind of see how they kind of go over and then down. And those would all eventually, a cable keeps getting bigger and bigger and bigger and bigger as more and more and more of these axons come together to start forming that optic nerve. So all of the information gets bundled into this one cable. So these are the axons, which are sort of like the cables that lead to the nerve. And that's why it's called. Eventually, it forms into this optic nerve with all of those axons put together. All right, hopefully this was a good introduction. Probably got a little more long-winded there than I wanted to, but hopefully that was a good introduction into the eye and retina. Thanks for watching.